Welcome back. So we're going to go for our last session for this second day. This is a session looking into rational combinations. So how are we going to move forward combining different treatments? Well, our chairs for this session need no introduction, really. We have Katie Kelly and Juan Vale, and I will just hand over to them. Thank you.
Thanks so much, Angela, and thank you all for being such great audience and so attentive and participatory throughout a long 2 days of meeting so far. But it feels like it's going very fast, and I, for one, have been so impressed by all the speakers. So it's my great honor to be one of the chairs with Juan today for this session, Development of Rational Combinations. It's something we always want to do, is combine an active drug with another active drug, but it's easier said than done. So it's my honor to present our first speaker, called Dr. John Gordan, who will be presenting on target identification to guide rational therapeutic combinations. John is an associate professor and physician scientist, MD, PhD at UCSF, and my clinical partner, and I've never ceased to be amazed by John's paired clinical acumen, as well as his incredible insight to all things related to protein structure and signaling. And I think he's the perfect person to talk about how we approach rational combinations in oncology. John's research uses systems biology strategies to discover new therapeutic targets and biomarkers for cholangiocarcinoma and other liver cancers. And with that, welcome, John.
Thank you very much, and thanks to the organizers for inviting me to speak. Because I'm the first speaker in the session, and because I think I am probably the most focused on laboratory science, I'm going to try to actually provide almost a preamble to some of the talks that are coming up and focus on how we can use actually some of the methods that Katie just highlighted to discover combinations. And so maybe let me just start by saying my disclosures, if I can get this to advance. Maybe a challenge. And if I can get help from AV, but I'm not able to advance right now. Oh, there we go, I made it, perfect. So I have in-kind support from Maradi and Effector, but now let me sort of get to this. And so I want to talk for a moment about just some work that came out of Peter Sorger's group at the Whitehead, which I found to be very useful when I think about how we use combination therapies. And I want to start by apologizing for this figure, which is ugly for several reasons, but I think illustrative. And it speaks to the distinction of how we think about combination therapies in the frontline setting. And then really for the rest of this talk, I want to make a distinction from that model from how we think about combination therapies in the later lines. So in the frontline setting, the combination therapies for cholangiocarcinoma and most other tumors remain combination chemotherapies. And if you look historically at the development of these regimens, it's not because of a mechanistic synergy between the drugs that are used, but rather based on the concept that's illustrated in this figure, which is that if one treatment, for example, gemcitabine, is able to help about 20% of patients, and a second treatment, cisplatin, is able to help another 20% of patients with only 10% overlap, then adding the two together, particularly for a tumor that's as aggressive as cholangiocarcinoma, is a rational strategy. The other thing that's interesting about the data coming out of Peter Sorger's group is it really illustrates that most of the combinations that we work with are additive rather than synergistic. And so what I'm showing you here on the left, or on the right, excuse me, it's my left, is that if you look at progression-free survival, which I think is a useful way to look at how these drugs work together, for a classic combination, for example, chemotherapy with trastuzumab, something that we see in breast cancer, the PFS curves actually, if added together in a model, overlap almost perfectly with a combination therapy. There are a few exceptions to this. Anything combined with an antivascular agent like bevacizumab, we tend to outperform additive. But I think for the rest of my talk, what I really want to focus on are therapies where we're actually targeting the same mechanism twice. And so these are things that are relevant in the second line setting. So what you can see here are the examples of a BRAF and MEK inhibitor developed for melanoma, but also used for biliary tract cancers. And so I'm going to talk about the lab-based discovery of these combinations and how they can be used to block acquired resistance. And then I'm going to talk for a few moments at the end about the work that we're actually very excited to take on in the innovation grant, using sort of almost reverse engineering the process, looking at active clinical combinations and looking to see if we can find mechanistic connections between the drugs. So before I go any further, I want to give a very simple primer, and I'm sure we've been talking, this is something that's been discussed again and again in different ways over the course of this meeting about how resistance to targeted therapy emerges. And I think in the new era of single-cell sequencing, it's been appreciated that most resistant clones to cancer or to cancer treatments exist at baseline. So if you're talking about an FGFR2 inhibitor, some of those point mutations that inhibit their blocking are probably present in any patient who's going to receive those drugs before they take the first dose. That said, a number of different things have to happen in the cancer for those resistance mechanisms to emerge. This is illustrated in this plot here on the right, showing how different clones are selected for under the pressure of a strong drug like an FGFR2 inhibitor, and bypass mechanisms that maintain survival during a strong inhibition are necessary for resistant clones to emerge. And so a lot of what we look at when we try to make combination therapies is to find those bypass mechanisms. I think we have to take a lot of lessons from lung cancer, which was the real prototype for receptor tyrosine kinase inhibitors and their use in anti-cancer therapy. So I'm choosing here a paper that covered the development of EGFR inhibitors from 2003 to 2013. It's not dissimilar to where we were a couple years ago with FGFR2 inhibitors. But what really stands out is from the first approval of erlotinib, which took place in 2004, within 1 year, the primary resistance mechanism had emerged. And only a few years after that, the bypass pathway was discovered. And then about 6 years after that, they developed compounds that mirror Relay and some of the other drugs, fudibatinib, that we're working with now for FGFR2, which are able to mitigate the appearance of resistance. Okay, so how do we discover these bypass mechanisms? Fundamentally in the lab, we have a couple of different tools. A very important one are functional genetic methods. So functional genetics, or sort of more colloquially CRISPR screens, are most commonly used to discover or to uncover a genetic relationship called synthetic lethality. This is the best example of synthetic lethality comes in the context of BRCA inactivation, where the loss of homologous recombination creates a sensitivity to disrupting other DNA damage pathways. But we can actually also use these kinds of screens to discover what we referred to kind of casually in a paper in 2018 as collateral dependencies. What are the genes or signals that are necessary to maintain tumor cell viability when you've inhibited a key oncogene? For the work that we did there, it was with KRAS G12C inhibitors, and we subjected both a lung and a pancreas line to what at that point was the most advanced tool compound. And surprising zero of us, what we found in fact was that the vast majority of the combinations, the genetic targets, most of which there were compounds for, that synergized with a KRAS G12C inhibitor were somewhere upstream or downstream of KRAS on the pathway. And that's exactly what we see with a number of other targets. It's important to note that in addition to combining CRISPR screens with small molecules, which is a natural way to discover drug combinations, we can also use combinatorial genetic methods. This can be done using sort of a number of different double guide strategies. The best one turns out to be sort of the oldest method, using two guides pointing towards each other to avoid homologous recombination. And this has been particularly useful in drug development because one of the challenges in functional genomics is that many of the enzymes that we target with small molecules have closely related paralogs. So for example, trametinib is an inhibitor of MEK1 and MEK2, and you're never going to find just MEK1 on a CRISPR screen because MEK2 is there as well. The other sort of key method for uncovering these combinations, of course, is multi-drug screens. And I'm going to highlight some work that we were lucky enough to be slightly involved with, but that really comes out of Nabil Bardisi's group, looking at infigratinib and fudibatinib, the two sort of key emerging FGFR2 inhibitors, and subjecting some of the models that his lab developed to combinations of those drugs with 111 other drugs. The other key point in this study is that they were focused on looking not just at growth inhibition, but the emergence of apoptosis, which we think of as a better way to stop tumors from growing. And, quite strikingly and similar to what we see with KRAS, most of the compounds that emerged that were, most of the compounds that emerged that cooperated with FGFR2 inhibition were actually EGFR inhibitors. Now, there's a natural toxicity question of combining these drugs together, but it really speaks to the biological relationships that underlie these combinations. And indeed, the Bardisi lab was able to show a marked benefit of combining even subtherapeutic doses of drugs targeting both EGFR and FGFR2. And so for the last couple of minutes, I want to talk for a moment about taking, instead of a screening strategy, almost a learning strategy. And what I'm going to present here is the work that we're hoping to mimic in a much smaller way for cholangiocarcinoma over the next year. So what I'm presenting here is a really impressive project that came out of a group in China looking at nasopharyngeal carcinoma. Oh my gosh, sorry, that slide didn't render. Let me go back just so it's not distracting. So nasopharyngeal carcinoma is a malignancy of the nasopharynx, as you might imagine. And as a result, it's relatively easy to get biopsies. And so in this study, a group of about 60 patients who were receiving gemcitabine and cisplatin, given exactly the same way that it's given to patients with biliary tract cancer, underwent biopsies before and after chemotherapy. And what was quite striking is that if you look at those patients who benefited from chemotherapy, those patients showed, in addition to chemotherapy-induced cell death, that there was an activation of antigen presentation, as you can see here, and an activation in particular of interferon signaling, both in terms of the expression of interferon-related cytokines and also the transcription that they elicit. This turns out to be mechanistically linked to cisplatin's mechanism of action. Cisplatin causes DNA breaks, which then activate an intercellular immune system called STING, which has actually been targeted therapeutically, although those agents are not particularly tractable for clinical use at this time. Looking at this work, they were able to find in those patients who were benefiting from just the gem-cis combination, the development of really profound immune complexes linking both T cells and B cells. And quite strikingly, looking now at a cohort of patients who went on to receive gem-cis pembro, mirroring exactly the standard of care for cholangiocarcinoma, or one of the two, I should say, those patients who demonstrated those immune aggregates did markedly better. And so just to sort of summarize what I've covered here so far, we think that combination therapies can prolong survival and I think are integral to how we practice oncology, either by helping more patients, so making sure that we have a drug that's good for more patients in the frontline setting, or by preventing the emergence of resistance. As we move into a precision medicine era, I think as opposed to trying to make more complex regimens to capture more patients, developing more targeted regimens to truly prevent the emergence of resistance is going to be the way that we can move forward. And as I talked about over the course of this discussion, combinations can be built based on either known pathway relationships, the BRAF-MEK example, or large-scale screening. And it bears noting that the large-scale screening often just returns those same pathway relationships. And finally, I just want to point out that we can do this backwards too, and that clinically active combinations can be studied retrospectively to understand optimization and potentially augment their synergy. And so with that, I just want to thank the members of my group and the people that we work with and all of our funders, and of course, thank you all for your attention.
I'm delighted to invite our next speaker, Dr. Funda Merick-Bernstam, who is the chair of the Department of Investigational Cancer Therapeutics of the Phase 1 program at MD Anderson Cancer Center, the medical director of the Institute for Personalized Cancer Therapy. She's a clinical trialist with a focus on decision support for precision oncology, biomarker-driven clinical trials, and antibody-drug conjugates. She's a physician scientist with a basic and translational research program focused on cell signaling, biomarker discovery, and molecular therapeutics, with a focus on patient-derived models as well as biliary tract cancers. Funda, we're looking forward to your presentation.
Thank you so much for giving me an opportunity to talk about combinations. I'm really passionate about this. And really, I think my talk is going to segue from in vitro target discovery to testing things in vivo and trying to decide what is worth moving to the clinic. Here are my disclosures. You know, we've had a lot of successes already in cholangiocarcinoma. We're so excited about our precision oncology efforts with the FGFR and recognition of RAF and IDH, and yet we realize that even our most effective agents have limited duration of efficacy, and ultimately there's a variety of different resistance mechanisms emerging, but highlighting the need to think about combinations. There are a lot of different strategies for combination therapy. You know, can we look at co-alterations that may modulate sensitivity? Can we look at adaptive responses to see how the tumor reacts when we give the drug so we can target that second survival pathway that emerges? Can we look at acquired mutations, or can we combine with things like standard care agents to enhance the activity or other molecular targets? I'm a big believer that trying to do the work these cells in vivo rather than just rushing to the clinic is worth the effort. And if we can do a better job, even when new drugs hit the clinic, before they hit the clinic, to identify biomarkers, maybe we can enhance the efficacy of each of our agents when we go to the clinic by biomarker selection. Even in the preclinical modeling stage, however, one of the things that's really important for physicians that are working with basic scientists is to talk about the clinical realities. What is really feasible to give in the clinic? Are two drugs too overlapping in their toxicity to make it happen? Will we be able to deliver the recommended phase 2 dose of both? Do we need to do that? Will we be able to minimize toxicity of both drugs by giving less because they're genuinely synergistic? Are we just giving more of one drug, less of the other drug that was efficacious, and ultimately not enhancing efficacy compared to the monotherapy alone? And if we're going to go to the clinic, what is the long-term goal? How would we register this? In what would be the clinical trial design be? And then what would be the patient population? So we really need to think about that so we can start designing our experiments in the lab to meet those needs. Also important to really think about in vitro, what's the signal? To be fair, a lot of things are synergistic in the lab, in cultures that are not showing anti-tumor efficacy even in the in vivo setting. So I am a big believer of in vivo work. However, even in vivo work has some challenges. First, in cholangiocarcinoma especially, we have very few models. We have a variety of different models. We covered this yesterday in the basic science session. They all have their own strengths. There's only a handful of cell lines. There are a handful of GEM models. There are a handful of patient-derived models. So we really need to work together to generate more models so we be able to really recapitulate what we see in our patients. And then we really need to think about how we would design experiments so we're testing realistic doses that are achievable in patients. We need to be able to test multiple models in vivo, which is challenging when we don't have multiple models. And we really need to have a higher bar. Many publications for every disease, but also in cholangiocarcinoma, you see success because there's statistically significant differences between drug-treated mice and the untreated mice, while the tumor's still growing. And as you know, in patients, we call that progression, and we take them off treatment. So we need to have at least stabilization in the mice, preferably regression. So in our effort, we've really heavily relied on trying to generate patient-derived xenografts for preclinical modeling. If there are any patients and advocates in the room, I just want to say our entire effort was funded actually through philanthropy in the space. So thank you so much. And these are models that are very valuable because you would not be able to capture what we're seeing, where we can generate models with a variety of different alterations. We can generate models that reflect the kind of patients we're going to treat in the metastatic setting. We're able to generate models from patients who have acquired resistance to different drugs. We can do these from biopsies. We can do these from peritoneal fluid or pleural fluid. There's some fantasy that maybe we can do it from blood. We're going to experiment. We'll see. They're doing it in Georgetown. And there's an advantage that this may be able to capture heterogeneity in tumors that we see. But even given the thousands of investigators that do research in vivo, there actually isn't clear guidelines about how to interpret in vivo experiments, how much efficacy you need to see, what is really clinically meaningful, how do you measure these. So there's actually an NCI patient-derived xenograft consortium, and I led a working group in this consortium. It took us 5 years to align on a manuscript that is coming out in a few weeks. I'm very excited. So, we have created a tool to give some guidance about how to interpret growth metrics in mice, but also a suite where you can interpret, put in your tumor model metrics so the calculations can be done automatically. And that, I think, also will increase the integrity of the work to ensure everybody creates something that everybody can sort of reproduce. And these are just kind of examples of how we do the growth analysis, growth curves, waterfall plots that represent what we see in patients, event-free survival that kind of mimics their Kaplan-Meier curve to see if we can improve survival. And importantly, when we look at combinations, it is so important to look at both drugs in the combination so that we can really see what has meaningful efficacy. I know these are figures that are hard to see probably in this room. It's also hard to see for me, but if you look at the first one that has a red dot, you can see that there's overlapping lines in the bottom. So if you didn't test all the monotherapy of each drug, you would get so excited that this is an efficacious combination. But in fact, there's one efficacious drug, and the second drug doesn't really help anything. In contrast, if you look at the models that we see in B, for example, you can see that both drugs have some efficacy, but in combination, there's a dramatic regression. And that's really the kind of thing that we're looking for. We want to be able to show that we're getting durable responses with combinations. I want to just give a couple of examples of the value of preclinical modeling from our lab. So for example, all 3 examples I'm going to give are related to HER2 because I think HER2 is such an interesting target in general, but especially for biliary cancers, especially in gallbladder more frequently. We had looked at in a clinical trial called My Pathway, we looked at the efficacy of trastuzumab in combination with pertuzumab. This was a combination that was effective in several tumor types. And you can see on the left-hand side is the waterfall plot from patients who had biliary tumors. Indeed, there was an objective response rate of 23%. The problem with this kind of a study is that we gave 2 drugs, and you really can't be sure whether you needed both drugs. And that definitely became an issue when there were discussions about whether this is a combination that can go to FDA approval. On the right-hand side, you can see that in the lab, we can test these things. So we took a PDX model that had HER2, actually mutation and amplification, and you can see that in this model, pertuzumab doesn't do anything. Trastuzumab barely inhibits growth, but the combination really enhances each other's activity. So I think it helps us dissect the value of a combination therapy when you have in vivo modeling. I'm also very excited about the idea of co-clinical trials. Can we get samples on patients going on trials and getting a biopsy anyway to generate patient-derived xenografts? So then we will have essentially an avatar of the patient, not to drive their decision-making, but really better understand how that drug works. We did this when we did the phase 1 clinical trial with zanidatamab. So this is a really interesting biparatropic antibody. That means The antibody binds the two sites on HER2, the sites that are bound by trastuzumab and pertuzumab. And you can see from the waterfall plot that this is actually a very active agent that had a 37% response rate overall in the expansion across a variety of tumor types. And in biliary cancer, the objective response rate was 38%, and we were so excited and rallied. And ultimately, as you know, the biliary cancer trial was conducted. But while we were doing the phase 1 study, every patient had a biopsy to look at whether they expressed HER2. So we were able to implant a biopsy at the same time to be able to generate a patient-derived model on each patient. There is a bias in what grows out. So in the upper two panels, you can see that patients' tumors, when we implanted it, if the tumor grew in the mice, the tumor was more aggressive. The patients whose PDX end up growing out had a worse progression-free survival, had a worse overall survival. However, still, we were able to generate PDXs across a variety of tumor types. And if you look at panel E, you can see that we had responders as well as non-responders with PDXs develop. So this is really a great mechanism because then you have PDXs that represent a variety of scenarios, including patients who had progression after initial response. So you can see that all 3 PDXs we tested after progression, after initial response, indeed had resistance in the lab as well. When we looked at the genomics of these patients, we saw that of the 4 post-progression PDXs, 2 of them had a MET amplification. You may remember the MET story because in the earlier talk, they were talking about MET amplification as a mechanism of resistance to EGFR. So we thought this was a rational thing as a mechanism of resistance for zanidatamab. And we went back and saw that indeed MET was amplified post-progression. MET was not amplified before the patient started treatment. And MET was not expressed before the patient started treatment, but it was overexpressed at progression. Then when we went back and started looking at cell lines, we saw that MET amplified or overexpressing cell lines were not sensitive to zani, even if they were HER2 positive, while those that didn't have that were very sensitive. Further, when we combined with a MET inhibitor, there was significant synergy. When we gave high levels of a MET inhibitor in these models, there was actually significant growth inhibition. And when we used a little lower dose of the MET inhibitor, you can see that there's enhanced activity with MET inhibition plus zani. So we're really excited about this finding, and we're looking to see if we can bring this into a clinical trial. Another drug category I'm very excited about is antibody-drug conjugates. You know, these are antibodies that bind the cancer cell surface that deliver chemotherapy. There's a lot of excitement in this space, and not only are there many ADCs that have shown activity in other tumor types, but also there's a lot of drug development in trying to figure out what combinations we can give. One of the big successes from this month is the antibody-drug conjugate targeting HER2. We did a clinical trial, DESTINY-PanTumor02, where we looked at trastuzumab deruxtecan. This is an antibody that targets HER2. We enrolled patients that had either 2+ or 3+ expression. And we saw that across the board, when we had 3+ expression, there was significant activity. And the responses were durable, so this was very exciting. This trial that I was lucky enough to be the coordinating investigator for led to FDA approval on April 5th. It was a pan-tumor approval, first time for HER2-targeted therapy, and the first time for an antibody-drug conjugate. Now, if you look in purple, you see the data for BTC. You can see that in patients that had 3+ expression on central testing, the response rate was quite high. That's the second bar. Now you see the third bar. Those were the patients that were 2+. You can see that there were no responses actually in that group. So really, we need to figure out a way where we can enhance the activity in patients that have low expression. So we've been looking at this low expression thing in the lab, and we've looked at it in a variety of tumor types. And I have to say that I initially started working on this in gastric cancer because many of my patients that were HER2 positive and I was trying to enroll them on trials, I realized also has cyclin E amplification. And we were looking at cyclin E as a predictor of response to a different drug class, that of WEE1 inhibitors. If you look at the second figure in the panel, you can see that when we tested low HER2 expressing but cyclin amplified gastric cancers, we saw growth inhibition by both drugs, but dramatic growth inhibition that was durable when we did the combination. We're very excited about this because we think this has implications for many tumor types, including biliary cancer that's HER2 low. And we're hoping to translate this to the clinic. This clinical trial is designed to start in the Experimental Therapies Clinical Trials Network this summer. Another NCI mechanism that is currently looking for combinations is that of NCI comma match. As you know, we're very passionately looking for arms for the master protocol for BTC, but just wanted you to be aware that there's yet one more trial ongoing that may have relevance for certain targets. I'd like to conclude by saying, these kind of efforts take a strong clinical team and really dedicated lab investigators. And I want to thank both the faculty and our investigators in the department, as well as our biliary tract program at Anderson. Thank you so much for having me.
Thanks so much, Funda. That was a fantastic talk. We'll hold all questions for the panel at the end. It's my pleasure now to introduce our third speaker, Professor Arndt Vogel, who will be presenting on rational combinations for FGFR fusions. Dr. Vogel is a full professor at the University of Toronto and is also a clinician scientist. He holds the Longo Family Chair in Liver Cancer Research and is leading a multi-arm research program in basic and clinical science at the Toronto General Hospital and the Princess Margaret Cancer Centre. And before leaving Europe for Toronto, he previously worked as a senior consultant at Hanover Medical School in Germany for over 20 years and still holds a research position there. Dr. Vogel's scientific focus is the translational and clinical research of gastrointestinal cancers, particularly hepatobiliary cancers. Thank you so much, Arndt.
Yeah, thank you very much for inviting me. It's a great pleasure to be here, first time actually, so it's good. Thank you. So BTC, I think, is really a very interesting tumor. I think it's very clear and acknowledged that the prognosis is very dismal, but I think in recent years we really have seen that we can make progress, and much is driven maybe not so much by the immunotherapy, which of course also contributes, but it's by the knowledge on the genetic alterations we have, and in contrast to many other cancers, many of these alterations are druggable as of today, and this is an updated figure from a paper we published last year, and the numbers are really increasing when we now think about the trials in KRAS G12D or with MTAP loss, as we have heard before. We have more than 50% of patients that have druggable alterations when we think about intrahepatic cholangiocarcinomas. In extrahepatic cholangiocarcinomas, it's a little bit different, and we have here three main drivers, I would say, p53, KRAS, and CDKN2A, which are not yet completely druggable, to say it at least. But we also have here a couple of druggable alterations which are a little bit different frequency compared to intrahepatic. But again, MTAP loss is very frequently, and also KRAS G12D is frequently observed in extrahepatic cholangiocarcinoma. So with the new drugs which are developed at the moment, I think it also makes a lot of sense to profile extrahepatic cholangiocarcinomas. And I think this is probably my first take-home message here, that we really need to profile these tumors as early as possible. We do have a lot of first-line trials. We have a couple of second and third-line trials, but also in the first-line setting. And when we would like to recruit these trials, we really need to do the profiling early. And this is what we have also mentioned in the ESMO guideline, which we published last year, which we are already in the process of updating because we have more approved drugs as of today, and we just heard about the tumor-agnostic approval for HER2, so this will definitely also be included. And I would like to focus on FGFR today, which is not the most frequent genetic alteration that is druggable, and maybe a little bit bit less than we have thought before. In around 10% of cases, we can find these FGFR2 fusions, which are not easy detectable. Again, this is the second point I would like. You really need to talk to your molecular pathologist to use panels that can detect these FGFR2 fusions, because I think it would be really bad to do profiling, but using a test that does not detect these fusions, because we have so many fusion partners, more than 200 have been identified, and you really need to employ tests that detect these fusions independent of the fusion partners. We do have a lot of drugs available. Sometimes I think there are more drugs than I have patients in my clinic. We learned today from a company that they're also bringing the next FGFR inhibitor on the market. It's a crowded place for sure, but the third point I would like to make here is there is consistent efficacy. We have disease control rates of 80%. We do see these nice waterfall plots, which is certainly exciting, but it's not all good because the responses are not yet durable, and I will come back to this in a second. The question is now, how can we make progress? And I think there are different ways how we can do that. One is we try to look what has been published and how can we divide intrahepatic cholangiocarcinomas. Different transcriptome profiles have been proposed, and this is, I think, a class that has been published 2 years ago, and I like it a lot. It basically separates the cholangiocarcinomas into inflamed and non-inflamed tumors, and unfortunately, these data support that the majority of intrahepatic cholangiocarcinomas are actually cold tumors, which is in line with the data we have seen for the phase 3 trial with checkpoint inhibitors. So we do see benefit, but it definitely could be more, and we are learning more about potential resistance mechanism. When you now look at these subgroups in the non-inflamed groups, we have the hepatic stem-like group, which is also enriched in genetic alterations like IDH1 and FGFR2 fusions, and we see a strong signature for M2 macrophages. So the idea could be that the drivers actually do not drive tumor development and proliferation, but also modify the tumor environment. And we have heard about some data earlier today. So the idea of a study we have started in Germany a couple of years ago was, can we combine targeted therapies with checkpoint inhibitors? And the idea was here that we use derazantinib, which is not only an FGFR inhibitor, but which has also CSF1R activity, which would allow us to target the macrophages. And the idea was basically, let's inhibit the driver, modify the macrophages, and then add a checkpoint inhibitor to activate the immune system. And with this kind of triple approach, we combined derazantinib and atezolizumab, and the study is still ongoing. I think 4 patients are still missing, and then we will get from the single-arm signal-seeking study, I think, some data to whether this would be a reasonable approach or not. The other approach would be, and this is what has been mentioned before, and I also like preclinical models a lot, and even if you look at the efficacy of the FGFR inhibitors and looking a little bit more to the left side, there are still some patients that are not responding. So having a driver does not guarantee success, and the question is, how is this primary and then later secondary resistance mediated? And to look at this, we basically used a preclinical model where we injected some oncogenic drivers, including FGFR2 fusions, and we were able to show that when we add the FGFR2 fusions, we get a very high penetrance of cholangiocarcinomas, which mimic the human tumors, and our control mix basically did not develop any tumors, indicating, okay, the FGFR2 fusions are strong drivers. So having now a tumor which is driven by FGFR2 fusions, we assumed they must be sensitive to FGFR inhibitors, but they were not. So the first step was, of course, telling the student, you made a mistake, do it again, and then we will see activity. So eventually I learned there was no mistake, and the cells were simply resistant to the FGFR inhibitors, and we thought, okay, why are they resistant? And we first looked whether the pathway is maybe not inhibited, but it was inhibited, so the drug was active, everything was active. And then we thought, okay, maybe it's about the co-mutations, and this could also be something that could be important for the clinic, right? And when we injected our DNA mix, we included also a mutant KRAS basically to induce the tumor. And actually, the mutant KRAS alone was not effective to drive tumor development, only when we combined it with FGFR2. So we need both drivers to that these tumors developed. And interestingly, then we asked the question, okay, maybe it's related to KRAS. So we treated these tumors with an shRNA against KRAS, and nothing really happened. So even though we need KRAS to make these tumors, inhibiting either KRAS alone or FGFR alone did not lead to success. However, when we combined the drugs, we were effective. And then we really saw in vitro and in vivo nice tumor shrinkage, indicating that activation of this MAP kinase, RAS MAP kinase pathway could mediate resistance to FGFR inhibitors. And as you know, and we have heard it before, it was not our study which really nicely demonstrated when we combine these targets, we are more effective. Also, a collaborator of us in Italy made the similar observation when we externally activated RAS signaling, we do see resistance, and if we combine, we can overcome resistance. And I think the paper from Nabil Badizi, which also was later published, also provided additional evidence that activation of this EGFR pathway mediates resistance. Yeah? And when we now look at more recent data that have been published, we do see that these genetic alterations also are detectable in patients that are refractory to FGFR inhibitors. So there's, I think, good evidence that this pathway is important to mediate primary and secondary resistance. And the idea and the concept, of course, for a couple of years now is to co-target these pathways. But the problem definitely here is toxicity. And when we combine with SHP2 inhibitors or MEK inhibitors, there's a lot of toxicities. Even the mice are suffering. So it's really difficult to bring this into the clinic, and we need to find different ways here. And the other point is, and then moving more to secondary resistance, is the observation. When we look at all the data that have been published, even the waterfall plots, they all look wonderful, of course, but we know that these responses are not long-lasting, and usually it's around 5 to 7 months, and then whatever target we look at, HER2, BRAF, KRAS, or FGFR2, we do see secondary resistance mechanisms. So this is something we definitely need to understand better, and here also a lot of progress has made for FGFR inhibitors, and I think you have seen this slide before at the conference here. So for FGFR inhibitors, specifically the first generation FGFR inhibitors, we do know that in many of these cases, specifically when they respond to the treatment, we have these on-target resistance mutation, which basically prevent binding of the inhibitor, and with that, the pathway is reactivated, and the tumors are progressing. So one concept, of course, is maybe we can just switch the drugs and use another drug that can still bind even in the presence of these genetic alterations. And so the question is, so when we have these drugs that are active against the resistance mutations, are they really the best-in-class inhibitors? So I think more and more drugs are developed as of today. Gordan, we have seen the data. But even if they are active in the refractory setting, they are not much better in naive patients. So even if they are active in patients with resistance mutation, it does not guarantee a prolonged PFS. So this is definitely something we always need to keep in mind, that there are not only these genetic alterations, but there are also a lot of non-genetic alterations, which I also would like to highlight here with this case. I mean, this was a patient we included in the FIGHT-202 study, and I think she was the longest, the patient that was treated the longest time in the FIGHT-202 study, almost 2 years, and she had a very nice response. Eventually, she developed progressive disease. She had an on-target resistance mutation. At the time, then futibatinib was available, so we started her on futibatinib, which was supposed to be still active and bind to the now mutant fusion protein. She again had a very nice response, but you can see the response was only for 11 months. Eventually, she progressed. Then she had a new resistance mutation, which was supposed to be sensitive to Relay, so we included her into the Relay study. Again, very nice response, 70%, third FGFR inhibitor. And then duration of response was only 4 months, and were not able to detect a new resistance mutation. So I think what this kind of illustrates to me that we can treat sequentially. We can look at the resistance mutation. They can indicate whether a patient might respond yes or not, but it does not really guarantee the same success. So the duration of response was shorter and shorter and shorter with each FGFR inhibitor, which we need to keep in mind. And then not only genetic, but also non-genetic resistance mutation kicks in. And it's usually not only one resistance mutation. Yeah, so this was a patient I'm treating in Toronto at the moment. He was included in the RELY study, and we did a liquid biopsy with him. And you can see it's not only one resistance mutation, but a couple of resistance mutations, and some of them at a really high frequency. So the liquid biopsies, I think, can really help us to learn more about the resistance mechanism to these inhibitors, not only genetic, but maybe also eventually non-genetic alterations. The problem is that sometimes we do not really are able to detect ctDNA in the blood, and I always wondered what's happening with the ctDNA? Why can we detect in some patients ctDNA and others not? And my understanding so far was because they're not shedding. So there is simply no ctDNA in the blood. But now I learned a couple of weeks ago, maybe it's a different mechanism. And I think this is really a very cool paper that was published in Science just, I think, 2 weeks ago, which basically asked the question, so what happens to the ctDNA? And actually it's taken up by Kupffer cells in the liver. And they identified a couple of nanoparticles or antibodies which they could block the Kupffer cells. And when you do that, you have a window of a couple of hours to basically increase the ctDNA. So this is still preclinical work, but they were able to show that they can increase the ctDNA in the blood by more than 10-fold. So, and I could imagine that this could be really helpful in the future, like contrast-enhanced imaging, to really improve the detection rate of genetic alterations in the blood of our patients. So looking very much forward to see this in the future. In terms of how should we best treat our patients, sometimes I think it's maybe also meaningful to look at like the cheap alternatives, even if they are not really cheap, right? But sometimes, especially now for me being in Canada, it's sometimes really difficult to get access to drugs. And one drug which is available, surprisingly, at least in HCC, is lenvatinib, and this was a study. And sometimes I think we forget that lenvatinib is really a good FGFR inhibitor, right? So there's clearly a rationale to treat also patients, cholangiocarcinoma patients with FGFR2 fusions with lenvatinib. And this is a study which confirms it, which really very nice in vitro, in vivo, and also clinical data showing that lenvatinib can inhibit FGFR inhibitors, and it also has activity against patients with on-target resistance mutations. So this could be something to keep at least in mind, and you have at least some evidence to refer to when you consider this in your clinical practice. Last but not least, being now in Canada, I need to raise this point. I mean, access to drugs, and we have discussed it this morning already in the session. So what are the challenges? And I think, I mean, we can think and do a lot about combination treatments and whatsoever, but we also need to make these drugs on a timely basis available to our patients. And this was a paper Angela and I published last year, and in this paper, we looked how long does it take from approval in Europe to reimbursement in a specific country. And this was in September last year. And you can see at that time, almost 3 years after approval of pemigatinib, there were still some countries in which pemigatinib was not available. At the same time, futibatinib was already approved 100 days in Europe. And you can see on the right-hand side, there was only 2 countries in which futibatinib was available, Germany and Austria, and not available in all the other countries. So the time from approval, from positive data to reimbursement and availability, even in Europe, can be quite long. And in Canada, none of these targeted therapies is available. And this is something that is really difficult for me, being for so many years in Germany, where we really had everything available, and now being in a country which I think is the first world country which has the resources to fund drugs like pemigatinib where they are not available. So we are trying to raise a lot of awareness. We are writing letters to the government, to the health insurance companies. And I think we really need to make this more public, and we need to come up and what has been suggested this morning. So how can we better do that? How can we better define what is really minimum standard of care for our patients? Yeah, and this was a study which I just found 2 days ago, where they really looked on the time of approval and reimbursement in Canada, and it lags behind a lot. So many drugs which are available in the US and Europe are not available in Canada. The time frame is more than 2 years, and I think this is something where also, and the Cholangiocarcinoma Foundation is already helping us, but I think we really need also patients involved in that we can really make these drugs accessible in North America, in Europe. I think the differences between the countries are still too much. So if we have the drugs not available, we need to invest in clinical trials. And this is, again, the good news, even in Canada, that we do have a lot of trials. And I'm really grateful to all the companies that invest in Canada and which basically bring their trials to Canada. And these are studies where we can participate at the moment. There are more to come. And I think the spectrum of genetic alterations is really important to acknowledge and clearly indicate that cholangiocarcinomas are tumors that require sequencing and druggable alteration. Thank you very much.
Thank you very much, Arndt. It's my pleasure to introduce our final speaker. Chemotherapy is not quite dead. There's still more to hear about it. And Dr. Masafumi Ikeda leads as Chief of Hepatobiliary and Pancreatic Oncology at the National Cancer Center Hospital East in Kashiwa, Japan. His expertise spans medical oncology, focusing on hepatobiliary, pancreatic cancers, as well as neuroendocrine neoplasms. Dr. Ikeda's contributions in patient care, research and education drive advances in treatment modalities and molecular understanding of these cancers. He's a respected mentor, educator, and advocate for cancer awareness, shaping the landscape of oncology with his dedication and leadership. Dr. Ikeda is going to talk to us about the development of combination therapy with cytotoxic agents. Masafumi.
Thank you, Juan, Katie. Hi, everyone. I'm Masafumi Ikeda, National Cancer Center Hospital, East Japan. First of all, I'd like to thank Congress Chair and Organizing Committee for inviting me to this Cholangiocarcinoma Foundation meeting. This is my first time to attend this meeting, so I understand this Congress was very active and informative. So when I go back to my hometown, I will recommend my colleague to attend this meeting next year. And also, it's my great pleasure to have an opportunity to make a presentation today. The title of my talk is The Development of Combination Therapy with Cytotoxic Agent. This is my disclosure. This is a history of the development of combination therapy with cytotoxic agent for advanced first-line biliary tract cancer. Gemcitabine plus, in 2010, gemcitabine plus cisplatin and gemcitabine plus oxaliplatin. So gemcitabine plus platinum doublet demonstrated a superior result over the gemcitabine plus or best supportive care. Gemcitabine plus S1 and capecitabine plus oxaliplatin. So fluoropyrimidine containing doublet demonstrated a non-inferiority result to gemcitabine platinum doublet. And gemcitabine plus cisplatin plus S1 triplet regimen and gemcitabine plus cisplatin plus durvalumab or pembrolizumab, also gemcitabine cisplatin-based triplet regimen, demonstrated a superiority result over gemcitabine plus cisplatin doublet. Unfortunately, modified FOLFIRINOX and gemcitabine plus cisplatin, nab-paclitaxel, was the negative result. I would like to talk about gemcitabine plus platinum doublet first. This is a phase 3 trial of GemCis versus gemcitabine, ABC02 trial. The study subject was advanced biliary tract cancer with performance status of 0 to 2. No prior systemic therapy were randomly assigned to a GemCis arm and gem mono arm. Median overall survival was 11.7 months in GemCis arm and 8.1 months in gem mono arm. Hazard ratio was 0.64, p-value was less than 0.001, so GemCis was a significantly better overall survival as compared to the gem mono. This is a Japanese randomized phase 2 trial of GemCis versus gem, BT22 trial. Study design was very similar to the ABC02 trial because of this trial is a bridging study to ABC02 trial. Median overall survival was 11.1 months in GEM-CIS arm, 7.7 months in GEM mono arm. Similar to ABC02 study, GEM-CIS shows a favorable overall survival. Third trial is a randomized control trial of gemcitabine plus oxaliplatin versus 5-FU fulvic acid versus best supportive care. GEMOX demonstrated a significantly better overall survival as compared to the 5-FU fulvic acid or best supportive care. Based on these three trials, GEM plus platinum doublet has been established to be a standard treatment worldwide. From this slide, from next two trial is a negative trial. First one is a Prodigy 38 and Amebica trial. This is a randomized phase 2 trial of modified FOLFIRINOX versus GEM-CIS. As you can see, the primary endpoint of progression-free survival were worse in modified FOLFIRINOX arm. So modified FOLFIRINOX fell to the better progression-free survival compared to GEM-CIS. Next one is a SWOG 1815 trial. This is a phase 3 trial of GEM-CIS plus nab-paclitaxel versus GEM-CIS. Primary endpoint was overall survival was quite similar between both arms. So addition of the nab-paclitaxel to GEM-CIS did not improve overall survival. So these FOLFIRINOX and GEM-CIS plus nab-paclitaxel was negative trial. From next two trial was a non-inferiority trial by the fluoropyrimidine in containing regimen doublet. First one is a Fuga-BT JCOG 1113 trial. This is a phase 3 trial of GEM-CIS versus GEM-S1. So you can see the Kaplan-Meier curve is quite identical between both arms. So GEM-S1 was found to be a non-inferiority to GEM-CIS because it does not require hydration. Next trial is a phase 3 trial of capecitabine plus oxaliplatin, CAPOX, versus gemcitabine plus oxaliplatin, GEMOX. As you can see, also the primary endpoint of progression-free survival was very similar between both arms. So CAPOX shows a non-inferiority to GEMOX in terms of the 6-month progression-free survival. Based on this trial, fluoropyrimidine containing doublet demonstrated to be non-inferiority to GEM platinum doublet. Next 3 trial is a superiority trial on triplet regimen. First one is a phase 3 trial of GEM-CIS plus S1 versus GEM-CIS, Mitsuba KHBO 14001 trial. The median overall survival was 13.5 months in GEM-CIS S1 triplet regimen and 12.6 months in GEM-CIS doublet regimen. Hazard ratio was 0.791. P-value was 0.046. So GEM-CIS S1 shows a significantly better overall survival as compared to GEM-CIS. Next one is a Topaz-1 trial. Phase 3 trial of the GEM-CIS plus Durva versus GEM-CIS plus placebo. Median overall survival was 12.3 months in Durva arm and 11.5 months in placebo arm. Hazard ratio was 0.80. GEM-CIS Durva demonstrated a significantly better overall survival. Last one is a phase 3 trial of GEM-CIS plus Pembro versus GEM-CIS plus placebo, Keynote 966 trial. This is a KT trial. So Median overall survival was 12.7 months in triplet regimen and 10.9 months in doublet regimen. So Gem-6 pembro demonstrated a significantly better overall survival as compared to Gem-6. Based on these three results, Gem-6-based triplet regimen demonstrated to be a superiority to Gem-6 doublet. This table shows a comparison of the Topaz-1, Keynote 966, and Mitzvah trial. The treatment outcome was slightly better in the Gem-6 S1. However, no big difference was between among three trials. So which regimen is best? Most important difference was cost. The cost of the drug is $12,000 in GC Durva, $6,000 in GC Pembro, $250 in GC plus S1. So the cost of the GC Durva and GC Pembro were 51 times and 24 times more expensive compared to that of the GC plus S1. Therefore, we are conducting to the phase 3 trial of GC S1 versus GC Durva or Pembro. Primary endpoint was overall survival. So at the present, around 50 patients were already enrolled in this trial. Next one is advanced second-line trial, second-line treatment. Positive result was FOLFOX and non-liposomal irinotecan plus 5-FU leucovorin. This is ABC06 trial, phase 3 trial of FOLFOX versus active symptom control. This is so median overall survival is 6.2 months is FOLFOX and 5.13 months in active symptom control. Hazard ratio was 0.69. So FOLFOX shows a significantly better survival as compared to active symptom control. Next one is a NIFTY trial. This is a randomized phase 2b trial of non-liposomal irinotecan plus 5-FU leucovorin versus 5-FU leucovorin. As you can see, the primary endpoint of the progression-free survival was quite separated. So non-liposomal irinotecan plus 5-FU leucovorin demonstrated a significantly better progression-free survival. So based on these two results, fluoropyrimidine-containing doublet has been established to be a standard treatment in the second-line setting. Finally, I will talk about adjuvant therapy for resected biliary tract cancer. A lot of the randomized control trial was conducted. Among them, positive result is a capecitabine adjuvant, and definitely positive result is adjuvant S1. First one is a BILCAP trial. This is a phase 3 trial of the adjuvant capecitabine versus observation. As you can see, the Kaplan-Meier curve was clearly separated. However, the hazard ratio was 0.81, p-value was 0.0897, so not significant differences. So adjuvant capecitabine shows a better survival, but primary endpoint was not met in the ITT population. However, the significant difference was seen on the recurrence-free survival and per protocol analysis of overall survival. Therefore, the adjuvant capecitabine has been regarded to be a standard therapy in many countries. In Japan, phase 3 trial of adjuvant S1 versus observation, a SCOT trial was conducted. 3-year overall survival was 77.1% in S1 arm, 67.6% in observation arm, Hazard ratio was 0.694. P-value was 0.008. So significant difference was seen in overall survival between two arms. Based on these two results, oral fluoropyrimidine agent has been established to be a standard adjuvant therapy for resected biliary tract cancer. So adjuvant therapy was established. We moved to the development of the neoadjuvant therapy. This is a study design of the neoadjuvant GEM-CIS plus S1 versus upfront surgery, JCOG 1920 trial. This is primary endpoint is overall survival. In the end of the March, the enrollment, the sample size was 460. So end of the March, total of the over 300 patients were already enrolled in this trial. This table shows a comparison of the guideline for the cholangiocarcinoma, Japan versus Asian country versus NCCN versus ESMO. In first-line treatment, GEM-CIS Durva, GEM-CIS S1, GEM-CIS and GEM-S1 was standard in Japan. Maybe GEM-CIS Pembro will be reimbursed in near future. On the other hand, GEM-CIS Durva, GEM-CIS Pembro, and GEM-CIS was standard in US and EU. In second-line treatment, no standard treatment was established in Japan. So maybe we usually use to the S1 monotherapy if it is not used in the first-line treatment. However, on the other hand, FOLFOX or nanoliposomal irinotecan was standard in EU and US. So adjuvant S1, adjuvant therapy is S1 is usually selected in Japan and Taiwan, and capecitabine was used in many other countries. This table shows a cytotoxic agent including standard treatment. In first-line treatment, gemcitabine or platinum regimen was frequently used, sometimes added to a fluoropyrimidine regimen. In second line or adjuvant setting, fluoropyrimidine-based regimen was often used. So gemcitabine, platinum, and fluoropyrimidine agent were key drug as a cytotoxic agent. This is a major ongoing phase 2 or 3 trial. Advanced stage, neoadjuvant stage, and neoadjuvant stage. Gemcitabine plus platinum and fluoropyrimidine-based regimen are under development. This is a median overall survival main regimen in first-line setting. Median overall survival around 11 months by the GEM plus platinum doublet regimen. And median overall survival was 12 to 13 months in GEM-CIS plus alpha triplet regimen. So doublet era has been changed to the triplet era already. However, the overall survival only improved by 1 to 2 months from 2020 to 2023. So in conclusion of my talk, gemcitabine, platinum, fluoropyrimidine were key drug as a cytotoxic agent for cholangiocarcinoma. As a basic regimen, these cytotoxic drugs, the development of combination therapy with immunocheckpoint inhibitor or molecular target inhibitor are underway. However, the progress of the cytotoxic agent was very slow. The development of more innovative drug is expected. Thank you for your kind attention.
Can we have the lights up, please? I'm sure that everyone is thinking about questions. It's a fantastic session on a kind of rational development of new combinations. Please come up to the microphones. I'm mindful that we're in a meeting with patients and caregivers, so I just wanted to ask Funda first, just to kick off. Can you please talk to us just about the importance of, you mentioned a couple of times, having repeat biopsies on progression. This is often something very difficult to ask when you've had a discussion around the cancer is getting worse, and why do you now need another biopsy?
Yeah, thanks so much. I don't know if it's working now. Thanks so much. I think we're increasingly understanding that the tumor evolves, and you know, there are many targeted therapies where we've seen emergence of resistance. Some are acquired mutations. There are some other mechanisms that really we don't understand as well, to be fair. So the value added of repeat biopsies is multifold. I think in a patient who's had a response with progression or prolonged stable disease, there's a significant likelihood that we'll identify a new emerging change that may help us target that with a new treatment choice. There is value added, no doubt, in liquid biopsies as well, and liquid biopsies can look at genomic changes, and it can help us also see what's in the entire body. When we biopsy, we identify change in only one area. But there are several also drug categories that require tissue. You may have heard me talk with much passion about antibody-drug conjugates. They are coming. They've revolutionized other diseases, and I have very much confidence in the next couple of years we will make a change in biliary cancer as well. To look at the targets for those drug class, we do need tissue. We don't know if the expression remains the same, so we're trying to look at that systematically. So I think sometimes we come ask for research purposes, for trials, we need to understand if the drug is working, if we inhibited the target. But often when we come after progression, you know, if we do a molecular test, it's also to help guide your therapy. So I think it's very important both for science and also potentially for care.
We have questions at microphone 1 and then microphone 8 or 3. Go ahead.
Okay. Thank you for the wonderful talk for all speakers. And I'm Yongye Su from Taiwan, and I have the first question to Professor Vogel. I was a little confused about the preclinical model about the FGFR and combined with KRAS. Because in clinical setting, KRAS mutation and FGFR mutation is basically mutually exclusive in intrahepatic cholangiocarcinoma, in my understanding. So I was a little confused about the model. You develop the FGFR plus KRAS mutation, because in my understanding, this mutation will not coexist in intrahepatic.
Yeah, I agree today. So when we started this project, we did not yet have these information, right? And that was one of the drivers why we looked at these 6,000 intrahepatic cholangiocarcinomas to better understand the co-mutational spectrum. Actually, when we initially thought about the project, there was a paper which was published actually in Journal of Hepatology, which suggested that KRAS and FGFR2 fusions would quite frequently could co-occur, right? So we were not completely opposed to that. We were a little bit hesitant to believe that this would be indeed the case, yeah. But at that time, when we started, we did not have the evidence to not use this combination. And actually, this was also, of course, one of the questions of the reviewers, and we also included other co-alterations, and we made the same observation. So we also used other activators of MAP kinase signaling. So it was not only KRAS, I mean, this was the driver of the whole story, but we were able to also confirm it with other co-alterations. But overall, I agree, we need to look very carefully at the co-mutational spectrum, and I think the preclinical models should should mimic the clinical situation as good as possible. And I mean, this was one of the reasons why we published the Journal of Hepatology paper last year, to really have a better understanding on which alterations co-occur and which not.
Okay, thank you. And may I ask one more question, a simple question to Professor Ikeda? As you mentioned in the first line setting, because there is no head-to-head comparison, but based on the data, we basically think GCS is basically similar to GC plus IO. So in the JCOC phase 3 study, which compared GCS versus GC plus IO, is that a non-inferiority design?
I'm sorry?
In the ongoing phase 3 JCOC study, which compare GCS versus GC, Durva or Pembro, is this study a non-inferiority design or is a superiority?
Yeah, non-inferiority trial. So that is a non-inferiority trial. So that's comparison is not head-to-head comparison. So that's cross-trial comparison. So you should see it with caution. But so the absolutely so the data was best in the GCS regimen. However, GCS is cost is very effective. So we selected the non-inferiority trial as compared to the GEM6 plus ICI.
Okay, thank you.
Microphone 3.
Hi, Sky Mayo from Oregon. A really thoughtful and engaging panel and moderation, so thank you. A question for Dr. Vogel and the group. We're at a point where we're seeing, fairly slim clinical benefits that are statistically significant. And I think one way to interpret the approval of some of the drugs in the European countries and maybe in Canada is perhaps a more rational utilization of resources. And I mean, for instance, I don't think that durvalumab is even approved for GCD in the Netherlands, because of the clinical benefit that is seen in that setting. So thoughts on what bar we need to meet in order to, push for these combination therapies. Thank you.
Yeah, I think that's a very fair question, and I also would not like to oversimplify this discussion. Yeah, and I think, the rational use of these therapies is probably really most important, and I think this is our task as community to come up with clear guidance on what we consider as clinically meaningful and effective, right? So if you argue that the resources are not there and therefore the drug is not reimbursed in a country, I completely agree with that. Yeah? And if you make this statement, it's too expensive, the value is not basically incorporated in the price. I understand. But saying that I do not see the evidence, because it's a single-arm study with 6-7 different drugs with disease control rates of 80% and response rates of up to 70%. To just state, I don't see the evidence because it's not an old-school phase 3 study with 2,000 patients, which maybe give you a hazard ratio of 0.8, benefit of 2 months, right? This is old school and I think not acceptable as of today, right? And we are talking about rare tumors, rare alterations, very small groups of patients, and we just need to come up with new ideas what we define effective. I completely understand, and I'm not, and I did not make the point that we should do it as in the US. I think it's great that you can do everything, right? I would love to do that, but I'm not sure whether this is the right way, and we need to be cautious with our resources, yeah? But we cannot just say we need a phase 3 study to see evidence. That's too simple. That's also not acceptable.
I just want to add one other comment that also addresses Masafumi's point about the cost of drugs. That when we look at combinations, we also have to factor in that the endpoint for one drug may not be the right endpoint for another. And when we combine immunotherapy in particular, we know that single immunotherapy agents don't always improve a median PFS or a median overall survival, but they are acting on the margins and creating a tail of the curve in some patients who have exceptional responses. And so I think while it's really important to pay attention to medians and the overall benefit in a large number of patients, we also have to pay attention to the durability of response and the exceptional responders that may have outsized benefit, really meaningful benefit at the tail of the curve. And so point being that median OS may not capture a population of patients that then go on to have complete responses or durations of response in the 2 or 4 year range. I'll draw people's attention to a poster we have from Topaz 1, which shows 3-year overall survival is almost over doubled in the patients on the durva arm compared to patients on the control arm because of this durability of immune response. And so one question for Masafumi is, if for the Mitsuba study, do we have long-term benefit, noting that the S1 agent is sort of our second-line agent that is now used up, and we don't have that durability?
Yeah, thank you very much. So, but in this trial is just only hazard ratio is important. So not so long tail effect is not so seen in this trial. That's trial. So yeah, that is a good point of the. So IO is specific to the long tail effect is a very good result. So what's Gemsys S1 is actually, so response rate is over 40%. And tumor shrinkage effect is very fantastic. And also, adverse event is also not so good, not so bad. So because of the GCS regimen is administered bi-weekly, so it is very feasible to the patient. So maybe in terms of the toxicity is maybe not so different in the GCIO. So we should focus on the cost effectiveness. So we select the non-inferiority trial, but we don't focus on the long tail effect.
Thank you so much. We'll go to Dr. Knox on microphone 1.
Thank you. Not so practical a question, but back to tumor models, maybe John or Funda. So you showed some very lovely examples of where it's quite convincing that one drug added to another is synergistic or significantly additive. But you know, in a lot of areas of oncology, we have multiple drugs. We're talking about triples and quadruples, and a lot harder to figure out in tumor models. And I'm just wondering, is there a detrimental effect that should be looked at as well? And could that ever then move into the clinic with a more simplified regimen? I know the answer, but I just want to explore the discussion a little more.
You take that.
I see you've given me the easy questions. I mean, I think that, so first of all, thank you for that very thoughtful question. And I think, I actually really appreciated putting this talk together because it gave me the opportunity to really sort of systematize how I've been thinking about drug combinations, which I'll admit was not something I'd systematized before. And I think we have to fundamentally distinguish between the combinations that are used to expand the capture of patients, the classic, 2 and 3 drug chemotherapy regimens, and the combinations that are used to extend the benefit of precision therapies. In sort of my own work and, things that I respond to very positively when I read them, I think that simplifying regimens is incredibly valuable. And I think that there's a natural kind of additive nature of oncology drug development that I think is relatively hard to circumvent. I think we have sort of rare examples that I can think of off the top of my head where things have been made simpler. For example, if you look at BRAF inhibitor development in colorectal cancer, they started, looking at 3 drug combinations with encorafenib, binimetinib, and some EGFR inhibitor, and then they backed off on the MEK inhibitor. So I think it's something that can be done, but I think I agree entirely. Perhaps bringing together both the patient experience and the cost effectiveness. I think being strategic about reducing drug burden is incredibly valuable, but is not something that we have the same models to develop as we have to find new combinations, even with incremental activity.
I agree.
Thank you. Microphone 4.
Hi. First, I want to say thank you to you guys for your work in this field. As a patient, I think a lot of us start out with one jaded oncologist that doesn't go particularly well, and it's nice to meet researchers and physicians who are interested in this work and have dedicated their lives to it. My question is, you brought up the need for additional biopsies. I feel like I remember in last year's conference, there was some conflict as to like what type of biopsy to get. I think you say percutaneous or the ERCP method, if I'm saying that correctly, and that there's some danger of seeding the cancer, or I just would love to hear your thoughts on that.
I think I wish we had endoscopists on the panel to comment on the ERCP, but I do think when we're talking about the repeat sampling, we are talking in the context of image-guided percutaneous biopsies, where you can see what lesions are growing on the treatment you're giving. So really do a targeted, knowing that if there's evolution, this is the most likely tumor, and also. Sometimes you can see that a tumor is necrotic, maybe it may not yield as good quality tissue. So those are the things where, a dedicated interventional radiologist that's really thoughtful about the value of the research biopsies is great to have. And there are sort of new technologies to try to look at the quality of the tissue when it comes out as well. But, definitely we really appreciate the partnership around these discussions.
Thank you so much. Back to microphone one.
Yeah, this is a question about the idea of maintenance chemo and how it may relate to, first-line GemCis durva. Taking into consideration the value of combination therapy, is there a value is there evidence or data that supports continuing GemCis beyond the 6 months that the Topaz recommends?
Maybe Arndt, do you want to tackle this? Yes, so a comment from me. When the ABCO2 regimen was first designed, it was very much time limited, and we accepted that after a period of time. We ran into issues around tolerability, toxicity, failure to recover of blood counts. So it was very much a pragmatic decision, knowing that we could then restart with some treatment at some time after that. We also know that in the Japanese study, the intention was for chemotherapy to continue indefinitely. So although the design was exactly the same, in that study, chemotherapy continued indefinitely. As you've seen, the results were the same. Now, we are revisiting this question. We're looking at some additional data from a number of international centers. And I think possibly the question now has been overtaken by the fact that immunotherapy is given as a maintenance treatment once you've managed to induce a response or disease stabilization with chemotherapy. That probably strikes the right balance in that you're still able to move away from some of the cumulative toxicities from chemotherapy, knowing that then you've got your immunotherapy then to maintain that response. And as has already been mentioned, that is improving that survival at 1, 2, and now we know 3 years as well.
So maybe if I could add here, so I think we can also acknowledge, and I think it's good to have this break of chemotherapy, right? I mean, after 6 months, I think it's also good to have like a chemo-free period. And I think it's maybe more important to not say it's now all over, right? So, but to continue to do imaging, look for tumor progression, but because we know that for some period of time the tumor is stable, and then to, as said before, to restart, right? So I would consider it as like a welcome break for recovery, and then but to continue treatment once the tumor is progressing, right? Either reinduction of GemCis or to use one of the second-line treatments.
Yeah, I want to thank everybody for staying till the very end of a long day, and took quite a while to put this panel together, so we want to take this opportunity. So the question I have for the panel is, with precision medicine evolving so rapidly and the tumor being quite heterogeneous from patient to patient to patient, we have many silos structurally, organizations, cooperative groups, countries, and just seeing all of you here, I can see that these silos are broken down, and we can have discussions that can maybe very quickly lead to advances. There's also barriers like getting things published in journals from initial observation, which could take years sometimes. So are there paradigms that you guys can foresee that can break down many of these barriers where knowledge can be exchanged much more rapidly, information can be disseminated rapidly, so that patients can benefit faster?
Who wants to tackle that one?
I think it's a one question. Yeah. That's a one question.
Perhaps I can tee you up with one short comment. So I think one very exciting thing that we're starting to see is the emergence of real-world data from de-identified EMRs. And I think that, each of us in our own practice at some time or another has obtained off-label drug for a patient with a targetable genetic allele that, doesn't currently, has strong support for another tumor, but not biliary tract cancer is, I think, a common thing that comes up. And I think being able to capture those data from large medical centers is one of the places where experiential data can be quite valuable. And I think the computational tools to extract that data are now coming online, particularly in terms of response definition and survival. So I think that's one sort of ray of hope to break down those silos.
Yeah, I think the other... Sorry, go ahead.
I think the cholangio community is really rare. Very few oncology communities have the collaboration, obviously the patient advocacy voice, but on top of that, I think the researchers and clinicians communicate and share data a lot earlier and a lot more openly than ever that I've seen in any other academic setting. So that does mean that we learn faster, and I'm hoping with activation of things like the master protocol, we would really be able to sort of get these preclinical signals and bring them into clinical trials much faster.
I would also like, I think we have really made a lot of progress in recent years, and I think it's really, it's a devastating tumor without any question, but I think we have made progress, right? And I think what we have clearly shown that it is feasible to make clinical trials in BTC, right? Like 10 years ago or 15 years ago, when I proposed an IIT to any of the companies, it was denied all the time, right? And now it's really, it's commonly accepted that it's possible, not only the pharmaceutical-driven trials, but also IITs, a lot of investigator-initiated trials. So I think we have clearly proven that we can make progress, that we can implement preclinical data very rapidly. What I think what is really missing for me is, and this is again my Canadian perspective now, is this: how can we bring this to the patient? The last step, right? So sometimes I think we are making maybe sometimes too many trials, right? So we are jumping too fast to phase 3 trials. We do another 100-patient single-arm study, but then it's not approved and reimbursed, right? So I think what we discussed this morning, the point that was made before, maybe it would be the rational approach to not reimburse all the drugs, right? Yeah, maybe it's true, but then I think we need to come up with definitions for that. And this is, I think, is missing at the moment, because with all these targeted therapies, and there will be more and more and more single-arm studies with more efficacy, right? We need to come up with definitions, what we define as effective, and then we really need to push hard that these drugs are available to all patients, independent where they are living, right?
So just to add from my point of view, alignment and collaboration. So alignment in terms of what we're doing, what data we're collecting, so that we accelerate rather than everybody's working in those silos. So you're absolutely right. But also aligning the priorities, and you already heard me talk this morning about making sure that we are all clear on what the priorities are for the community so that we can all move in the same direction. So thank you for staying over time. There are still 2 people at the microphone, so we will take these questions. Please stay. Everybody's very engaged in this discussion. Microphone 4.
Following up on the earlier question about stopping the GEM-CIS after 6 months and continuing the Durva, for patients who then move on to a second-line treatment, a targeted therapy or F5FU or something else. What about continuing the durva then?
I think we will need to have data for that. I think, I mean, in clinical practice, I do hear it quite frequently. So should we continue beyond progression with the immunotherapy? I think we do need the evidence. In hepatocellular carcinoma, we had the same questions, right? I think the HCC is responding even a little bit better to immunotherapy compared to BTC. And we have now initiated phase 2, phase 3 studies to exactly ask this question. Can we and should we proceed with the immunotherapy beyond progression, right? And I think we have seen for Masafumi a very good example on the costs, right? And again, I mean, this comes together again, not only toxicity, but also financial toxicity, right? And I think when we are talking about these costs, we really need to provide the evidence that it's meaningful to proceed. But I think it's a worthwhile question for sure.
Yeah. And so is that a sort of trial we can expect to find?
I think it would be good to have a rationale to do that. Yeah, I mean, I think in HCC we have combined everything with everything. It did not always work out, right? I think it's always good to have a rationale why it would make sense. Yeah, I think we do have some preclinical evidence suggesting that TKIs like lenvatinib maybe modulate the tumor microenvironment to immunotherapy, so it makes sense to use it after bevacizumab, which has a different mode of action, right? Just continuing without any rationale, I would be a little bit hesitant, and I would prefer to have a rationale to do that.
There are phase 3 trials going on in HCC, hepatocellular carcinoma, to answer that question, and I think that it's important to highlight the CCF's prescience in hosting or calling for the immunotherapy resistance proposals to study exactly what is the mechanism at hand, because without knowing the mechanism, it's hard to support continuing indefinitely a drug that has a lot of toxicity potentially over time.
Thank you. Microphone 3.
Yeah, you can hear me? Okay, there we go. So you leave me in the precarious position of standing between a room full of people and their dinner. So I'll try to make my question worthwhile. So my question is about sequencing, and one of the most fascinating aspects of this conference is getting the input from the patients and from the caregivers. And one of the most surprising pieces of feedback I got from a patient, and from a few of them, was that they wish that they had done the clinical trials before the first-line treatment when they were in their healthiest position. And so their perspective was, we're healthiest now, let's take the riskiest approach, and then if we're not doing well, then let's do the standard approach. So that leads me to asking 3 questions. This is a combination permutation question, which is, first of all, for the first set of treatments, what are your thoughts on doing either clinical trials or second-line treatments that sometimes have more promising long-term results? And then, in terms of if the first line doesn't work, what are your thoughts on skipping second line and going to clinical? Because there were some doctors earlier in the sessions yesterday that said, look, second lines don't have great outcomes anyways. You know, the FOLFOX has a 5% survivorship rate, so you may as well skip that and then just go straight to clinical after first line. So the first part is, what are your thoughts of not doing first line and doing a clinical or a second line? And then if you do first line, what are your thoughts on skipping second line and going straight to clinical?
I can address one part of your questions, maybe. So I think it's probably worth to think about going to earlier settings. I think there are some practical obstacles when we talk about profiling, for example. It usually takes some time, and we have seen that the phase 3 studies with FGFR inhibitors basically never recruited their patients, so it's really difficult to include patients into first-line trials based on NGS reports. So this is just a practical issue, but I think we can make progress on that, and it should be feasible, and will see more trials in the first-line setting in the near future. Another approach would be to go into the neoadjuvant setting, and this is something we are just seeing in the Artomite study, for example, where a bispecific antibody is investigated, which has not been tested in later lines, but in the neoadjuvant setting in the adjuvant setting to understand efficacy. So I think there is there could be some rationale to use these drugs in earlier settings. Sometimes it's practically difficult, and sometimes it's always also depending on the risk, right? Because we know that some patients are really progressing very fast, and if you have a drug with a low level of evidence, you may miss the opportunity to deliver evidence-based effective therapy. So it is like in pros and cons, yeah, and we always need to balance risk and benefit, but I understand your point.
You know, clinical trials are written, you know, for specific patient populations. So, you know, it's usually not that somebody can go on first line or second line. We really need to have efficacy signal for us to have the confidence to forego an effective first line therapy. So current first line trials, there are trials in the first line setting, are building on something that we think is already effective. And we're just trying to figure out if it's better than what we have as an alternative. So of course, if a patient's in good enough shape and motivated to go on first line, we're very, you know, supportive of those first line trials accruing, because we think those trials are hopefully going to do better. That's the only reason we're designing trials. In the context of second line therapy, indeed, I think we're all kind of disappointed about what we have second line. So I think many of us would be very supportive of going on a clinical trial in lieu of what we have standard of care second line. But it's also important to know what is the trial, what is the scientific rationale, what do we know about this agent, so you can really make that decision with full informed consent.
Yeah, I was just going to add a comment that you know, we only really know a drug is effective until after the trials happened. And so you have a group of people, a body of people who are fighting your corner. They're called the ethics committees. The ethics committees want to make sure that if we're doing clinical trials, it is ethical to do so. And so if you have a drug that looks really good on paper, looks really good in the lab, you still have to have a little bit more evidence to suggest it might be effective. If you're going to say to a patient, I'd rather you don't have the standard of care treatment, try this trial first, the ethics committees would push back against that. They're saying you're compromising something that you know works. It may not be brilliant, but you know works for something that really isn't very tried and tested. So for that reason, we have to gradually bring these things through, get as strong a signal as possible that drugs are going to be effective, and then work with ethics committees who challenge us, quite rightly, to make sure that we're using the drugs at the right time without compromising patient care and safety.
Thank you.